Thank you, thank you. Oh, now there's magic of my voice. Thank you so much. So much better. So yeah, software engineering, 2011, master's. Actually did my exchange at Royal Institute of Technology in Sweden for a semester, but then came back. I genuinely think that Tartu is a great place. Really loved it. Stayed here. I was invited by Jaak to go into PhD. Never regretted ever. I really loved the process. What Jaak said about pivoting was a very polite way of saying that I was a social butterfly that liked to jump around from nice project to another nice project. So in that way, I did some sort of like non-invasive tests for cancers. I did research around self-driving cars with autonomous driving lab here in Tartu with Tambet Matisen. And of course, the research that I'm going to be talking today. It's all about this. Now, a little bit of expectation management from my side about this talk, about this. It's more about lecture, it's not so much talk. It is about a vast range of things that we have been working on, but also other people have been working on in computer science in relation with medicine and biology. So this is the topic of the talk, is how can people with background in computer science, such as ourselves, contribute and do things in medicine and biology. So that is going to be about it. Also, my focus specifically is going to be on imaging modality. So I'm working with images. People I work with usually work with images. So it's going to be all about computer vision. I know that some of you, or maybe most of you, don't have background in computer vision. So I will try to stay either brief and shallow on the computer vision heavy things, and I will also try to give you as much background and as much context when it comes to computer vision when we cover these topics. So yeah, without further ado, usually when I present this talk, and I have presented this talk already for some time, although this is a new version of it, I would like to explain the complexity that people that work with medical and biological sciences, what kind of complexities they're facing. And for this, I usually try to anchor people into thinking about some very vastly large number, like huge number. And to do this, to facilitate the process, I usually ask them a question, leading question, such as, how many do you think there are stars in our galaxy? And please give me ideas, like what do you think, how many stars there are in our galaxy? Millions. Billions, yeah? 200 billion, roughly that, right? When people are sitting next to computers, it's obviously easy shortcut to find this answer. But basically, you're right. So it's about 200 billion stars in our observed galaxy. Now, let's switch the gears a little bit. And I will ask you another question. How many do you think there are cells in an average grown human body, such as mine? There is nothing special about it. It's fairly average in all the kind of ways. What do you think, how many cells there are inside of this package of flesh? How many cells? Dima, how many cells do you think there are? In couple of trillions, yeah, about that. Fewer, more? What do you think, guys? More. How much more? How much less? More than a million, right? That's a safe bet, very safe, especially after somebody said trillions, right? Chingiz, what do you think? How many there are cells in that package? Couple of trillions? Okay, I will not torture you anymore. It's about 37-something trillion cells. Depends on the dimensionality of that package. It's about 37 trillion cells. So you can see that we actually have to shift the focus a little bit from stars to bodies. Also, my personal preference is on cells and bodies because stars, if you think about what are those stars that are glowing in the sky, these are heavily packed, usually uniform matter. It's packed so densely that it starts to shine, it starts to emit light. right? While our bodies are very complex interplay between various different molecules and substances that communicate in vastly complicated ways to make what you are seeing right now talk and walk, which is beautiful and amazing, right? It's still the same stuff packed in, but it walks and talks. It's great. So our bodies and our, what we are very complex creatures. So we are made up of trillions of cells, right, which make up tissues, dozens of tissues that make up organs. And these organs actually make us who we are. They make us live. They enable life in our bodies. Now, as I guess everybody is well aware, there are lots of things with these complex systems. When we are talking about very complex systems, such as this system, it's very easy for complex systems to harbor some sort of small issues that sometimes accumulate and become big issues that break the systems. It's very much the same with us. There are lots of ways how our complex systems can break. But there is one very common and very much popular and talked about way how this sort of systems break. And I think most of you know what kind of story am I going to tell you. It's a story about cancer genesis. We call it cancer genesis is when cancer starts, right, appears. And it starts by a rogue cell. There is a rogue, rebellious cell. It's not a bad cell, it's just the one that does not go along with all the others. This cell acquires certain types of mutations in the DNA, and it starts to reproduce chaotically and massively. It starts to create copies of itself. It starts to become invisible for immune system, which normally kills that sort of type of cells, because we all have that sort of cells. Even right now, someone is developing that sort of cell somewhere in their bodies. But these cells, very hopefully, I'm very hopeful that they will be detected and destroyed by the immune system. But this one was not destroyed by the immune system. So it accumulated. It started to create more and more copies of itself, and it created a hub of like-minded cells in the tissue, which actually manifests as a tumor, as a bulk of tissue somewhere in the organs. This bulk of tissue grows in, and it starts to dispatch sort of like representatives all across the body. It sends the representatives all across the body, and these are called metastases. And these metastases, they establish colonies in far organs, and they start to grow as well. And so it becomes basically this sort of like a growing mass of stuff in different places, and it eventually leads to obvious death of the host. So why am I telling this, right? There is a very fair question usually from the audience of the computer science background. Why are you saying this sort of things? Now, thank you. I harbored another fear in my life. Thank you for doing this. Why was that even useful? What am I trying to say? What am I going to say is that actually many things, and you have seen that this sort of process has multiple levels, right? It starts from the cells, goes into tissue, and then goes into organs. So on many levels, there are certain processes that we can help either analyze or build solutions for. Meaning, when I say we, I mean people with a computer science background, those sort of dudes with the laptop, right, on the one side of the slide. So it turns out that there are things we can do that can help to solve problems on the other side of the slide. So what are these things? So let's start with just an idea of what are the things this kind of guys with this sort of nice and shiny laptops like here can actually have been very good at. And we have been very good at certain things. So what are these things that we have been good at? We have been good at building AI systems that can classify millions of images. We have been pretty good at, let's say, controlling, or let's say, mimicking human drivers' behavior from the camera feed. We were able to say where to steer, when to brake. We were pretty awesome at creating models that can mimic human ingenuity and imagination. Obviously, you're all well aware that there are engines that we have built that can write text as if it was a human, going through the Turing test and many other things. So it turns out that many of these things that you can see on the left have a direct application to the things that I presented about the human body and cancer story. And I will tell you, so in this talk, I will attempt at showing you which specific problems can be solved, and so that you may actually think of other ways, or maybe your ways, or maybe some other applications that are possible using modern AI, and maybe something that you can actually help build in the future. I will give you, so right away I'll give you a disclaimer that many of these things that I'll be talking about, they're based on what my research group, which we call Biomedical Computer Vision Lab, have been working on. I will be also naming some people that were behind some of the projects, but also I will be trying to give you a more broad picture of what is in general out there that you yourself, maybe your collaborators, your colleagues could actually participate in. So let's start with the very first layer. So you remember we went from cells to tissues to organs, right? Now let's start with the cellular level of cells. What you're seeing here is actually cancer cells under a microscope. These guys are cancer cells. These specifically cancer cells have a name. They're called HeLa cells. Some of you, I suppose that some of you might actually know what HeLa stands for. Any immediate answers? Yeah, because I told you. Wait. HeLa stands for Henrietta Lacks. This is a woman who lived in the United States in the midst of the previous century. She died of cervical cancer. So she actually died of cervical cancer. Cervical cancer is basically a women's disease. It's affecting reproductive organs. It's a very aggressive type of cancer, one of the most aggressive types of cancers. So she actually died of the cervical cancer. And these are cervical cancer cells that still live on. Almost 100 years after her death, they're still alive. And they're called immortalized cell line. Immortalized means that they will live on for as long as you feed them. They also like to eat stuff. They like glucose stuff. So if you feed them and you keep them relatively safe, they will live on. And this is called immortalized cell lines. Why do people need immortalized cell lines? They serve as a role model or model of a cancer. So cells in general, in biological research and medical biological research, they serve as a model of how cancer would react to a certain chemical that you, for example, let's imagine that you have developed a new chemical that you want to stop cancer. What you do is you don't go around and do this and then put the syringe into somebody's arm and say, oh, let's check. Now, please, sorry, I have to observe you for another 5 hours. First you start with cells. You grow those cells, right? And then you put this chemical and then you see what happens. Well, this is a very simplified version, but that's roughly what it is. And obviously, if you're building a drug against cancer, you would like to see those cells die horrible death, right? Because, well, we are trying to kill them after all. So how do you know they died? That's a pretty difficult thing, right? You have to look and check whether they are alive or dead. There is a problem here. There are too many of them. They're very tiny. They're cells, after all. You can't see cells with your bare eye, right? So we have to use microscopes. There are hundreds of thousands and sometimes millions of those cells living on those small petri dishes. So how do you know some of them died, some of them didn't die? Did your drug make any difference? You have to run something called a large screening study. You have to look at those cells without your drug and with your drug, and then you have to calculate how many of them are alive and how many of them are not. And if there is a statistical difference, you can conclude, yeah, my drug has worked out. I see one question here. Go ahead. I will just repeat it because I can repeat the question. It's perfectly fine. Can you please repeat it into the microphone, because otherwise the audience from online or whoever is watching the recording will not be able to hear you.
So if cells are dying, I mean, it will reduce the amount of reproduction of the cells that would happen. So can't you just see it by seeing the overall decrease of the reproduction of cells that would happen within the sample?
Right. So the question was about the reproduction. It could be that the cells will stop reproducing, right? So your drug may not necessarily kill the cell, but it might cause them to just not reproduce, which would be also a favorable outcome, right?
Correct.
Yeah, but at the same time, you still need to count them. Basically, you have to figure out if they're still reproducing, it means that they would double in size or double in quantity, right? If they don't reproduce, they would just stay the same. So this leads us to the same very problem. We have to count the cells, right? So we have to build algorithms that are able to look at these microscopy images. There are other ways how to count cells. This is not the only way to count cells. There are a lot of so-called wet lab ways to count cells. But this is one of the most, so far, affordable ways to count cells. You make images, and then you count them. But of course, there is a problem. There are many of them, so we need to automate this process. So what people have been doing, and we also have been doing a lot, is building such algorithms that look at microscopy images, and then they say, all right, there are so many cells here, and there are this many cells in here. And then we can compare, right? This is one stream of applications. It's counting cells, detecting cells. Now, yeah, this is what basically it says. This cell line that I have been showing you, these cells, the microscopy image itself is called a fluorescent image. Why is it called fluorescent image? Is that it emits light. You can see the cells. You can see where the so-called nuclei of the cells are, where the main parts of the cells. They shine bright at your eyes. You can see them. Now, try to take a look at these images and try to find cells here. I have actually skewed the contrast so you can actually see the outlines of the cells. But normally it's a gray nothingness. It's very hard to see through anything there. So these are called bright field images. And one of the challenges was that in fluorescent microscopy, the previous type of images that you have seen, you have to use a certain chemicals. They're called dyes in order to basically see the cells. These chemicals are expensive. Somebody has to do the experiment. And also they kill cells by themselves. You don't need drug. You just put the dye into it and they disappear. So you can't really observe those cells for longer periods of time because these chemicals, they kind of mess up with the cell cycle. So you can't really do it. Now instead, and yes, if somebody had the question, why don't we have then use them as a drug against cancer? The problem is that they're toxic to every cell. So you would die with the cells. You don't want that, right? So you want very specific things to die. These images, on the other hand, are very cheap to acquire, meaning that you don't really need to pay anybody. You just, have a microscope and you just do chuk, and that's it, done. But they are very hard to analyze. So far, before the AI kind of technology entered the scene, they were considered to be rubbish. They were considered to be basically a byproduct. They weren't considered to be anything useful. But it turns out that you can use the same sort of AI models to detect the same nuclei now from much more complicated type of modality. This is something that has been the most maybe fascinating about the whole AI thing is that humans cannot do it. We ourselves, by looking at the images, we very rarely can see the actual outlines of those objects that we are interested in, but the model can find it. Model has infinite capacity of patience to go through the patterns, analyze large amounts of data, and then actually detect those objects on those images. So we have been doing that as well. Now, moving up a little bit up the ladder of complexity, going from cells to tissues now. When it comes to tissues, there is one very important way how people are usually diagnosed with cancer, and it's called biopsy. I hope that none of you know what biopsy is. Okay, so there are some people who know what biopsy is. I hope that it's not your personal experience, though. A personal experience. I'm very sorry for that. Basically, biopsy is a small type of surgery when usually a surgeon puts a needle into a human and into a tumor. They snatch a sample, a part of that tumor, for the analysis. Then they take it out, they slice it up, and take images under the microscope. Why they do it is they want to check certain characteristics of those tissues. For example, as I said, remember that the rogue cell, it multiplies endlessly. So if that tissue under the microscope looks like lots of multiplied cells, then we know for sure there's malignancy, potential malignancy in question. So basically, malignancy is this property of the tumor to metastasize to other organs. This whole process that I have described is manual work. A lot of manual work. It's these images that they get from this tissue slices. There are dozens of thousands of pixels in size, the resolution. So it's vastly large images. Each image can weigh up to gigabytes and many gigabytes. It's very complicated work that they do. So we have been building actual models to try to optimize this whole pathology, digital pathology workflow, starting from the images and actually helping them to figure out what the diagnosis could be from that image. So for example, by the way, this image, I kind of lied to you. It's not a cancer cells here, but these are male testes, and the condition is infertility. So this was actually a long time back research on infertility in men. And basically what you're seeing are the tubules. These are certain structures in the testes that has to have enough type of cells for a man to be able to have a child. And then you have to calculate cells, and there are all sorts of parameters where you have to calculate the fractions of different types, and these fractions help actual doctors to make actual diagnosis. You can imagine that counting the cells within the tubules, finding those tubules on images which have like dozens of thousands of pixels across is enormous work. So we have been building models, AI models that can detect those tubules, that can segment those tubules, and that can detect those cells within the tubules, and then can actually help to move on with the diagnosis faster. Moving up the ladder again, now we are at the organ level, right? So this is for me, it's the final layer, final level of that hierarchy, going from cells to tissues to organs. 4 years ago, I have co-founded a company that is called Better Medicine. Better Medicine's goal was to build, and is still, build AI models that can actually help detect tumors when they are already detectable from the various imaging modalities such as computer tomography and magnetic resonance imaging. So what we are doing is we are looking at this sort of CT scans, and we are interpreting them using AI. We are helping radiologists, we are pre-doing their work, we are, in a way, basically we are formulating our task as we are building a co-pilot for radiology. We are building a co-pilot. We are building the second pair of eyes for radiologists. For example, what you are seeing on this picture. What can you see on this picture? Yes? You see kidney. You actually see two kidneys. One is a little bit like starting, right? It's not yet there. What else do you see in this picture? So somebody said kidneys. This is very advanced, very much advanced. Easy things, easy things. You see the backbone there, yeah, you see a like white backbone, so this is right in the middle, the backbone. What are these white spots? Miriam, what are these white spots here? What do you think are this? This is a backbone. What do you think are these white spots here? These are ribs, right? These are ribs. This is a kind of section of the ribs. But what you don't see, likely, right? First of all, you probably don't see a liver here, but it's there. And what you don't see is a huge pancreatic tumor. It's actually a big one here. It's a huge pancreatic tumor that just looks in the eyes of the observers. Obviously, so don't take me wrong. Any radiologist who would ever wander into that presentation, they would say, well, I knew that right away. I mean, it's so easy. But there are circumstances when we are helping them to see those smaller things which sometimes hide from the plain sight. And we are helping them in being this sort of like a safety net. We are providing a safety net. Sometimes they're unsure. Sometimes they want to, have some second opinion. So that might be one way to give the second opinion. We are quite proud because very well recently, a bit like more like several months already, but we have received approval from the European Union to sell the very first commercially available AI model that can detect and measure the kidney tumors, kidney renal cancers. This is basically the very first commercial product for this sort of use in the entire world. There's no other kidney tumor detecting AI out there, so we were the first ones. Well, it does have to say something about the necessity and urgency of that, but we're still being very proud of that achievement. Right, so just to kind of briefly go around what I just said about different levels is that we are working on a microscopy image level, on a level of cells, to try to detect cells, usually cancer cells. We also quantify different parameters of those cells. Remember when I told you that you're developing a drug against cancer, you would like to see cells die in a horrible death? Now, what death is an ultimate goal, but you are also happy if they get very stressed, if they get very unhappy, if they scream out of pain and that sort of stuff. We're also happy about this. So you can't really see that. Beyond that, you have to measure their sizes, morphology. When they're stressed, they become star-like, they squeeze, they don't like stuff. When they are happy, they become big and round, and they love that sort of things. So we are also measuring this. We are not just saying this one cell, this is the other cell. After we apply our algorithms, you can also analyze many sort of properties of those cells. Then on the second level, we were having projects with local hospitals where we analyzed pathology images, so these pink ones. We were building algorithms, and I actually had a few students defend it with their master theses around the topic of basically automating digital pathology workflows. And finally, on the level of organs, we are working with my company, Better Medicine, building various different AIs around detecting tumors in different organs. We have moved on since this big kidney announcement. We have moved on. We have already looked into lungs. We have looked into pancreas. We have looked into liver. We have started to look into also lymph nodes and bones. So we are really trying to cover the entire internal organ systems. So we are not looking into one organ in particular. A kidney was just a starting point. We actually want to cover the entire thing because that's what good copilot will do. This is what the good assistant will do. Not just look at the kidney and say, I have your back, everything is cool, but actually going through the whole thing, right? Because the small little lesions can be missed, and we don't want this to happen. Okay, and now now I know that I have approximately an hour to go through the rest of my 100 slides or something, 120. I get the question right now, but before I get the question, I wanted just to say that I am going to show you as much as I can in the remaining time from the research that we have been doing on different levels. And I'm very sorry if I won't have enough time to cover everything, but I'll do my best. Sorry, go ahead.
Thank you. Very nice presentation so far. Can you comment on that? In the top left, you had that Revity and Syngenta. What are those?
Yeah, sure. I will mostly talk about them in more detail, but basically, Revity is the company with whom we have been developing many of the microscopy-related technology. So they are our collaborators. They have provided data on which we have been building these algorithms. They have also provided a problem description. So many of these things that I'm going to be talking about in the cell level, they come from them. They are interested in this. They are producers of the microscopes that many labs around the world are using to study cancer. So things that we are building might and have already ended up on the tables in the actual lab workflows of practitioners around the world. Syngenta is one of the companies with whom we also collaborated shortly on a certain project related to microscopy. I'm going to be talking about them as well. Also, you can see labels and logos of Ida-Tallinna Keskhaigla and Tartu Ülikooli Kliinikum. These are two hospitals in Estonia. One is in Tallinn, another one is ours here in Tartu, with whom we have been collaborating when it came to pathology images. We are still working with Tartu University Hospital here when it comes to also better medicine work. So they are very integral part of this whole research. Okay, let's move on. So basically, as I said, I will try to cover the most juicy parts of the research we have done with all these levels, with different levels. And I'll also try to keep it in a way general enough that people with general computer science degree and background would still follow. I will start with this idea of detecting cells from bright field images. So this was a big thing back in 2017-18 when we started this work. And what I can say is that, well, yeah, you can look at this image and then you could see the overlay going back a little bit. So this is bright field image. And now you can test yourself whether you are able to see the nuclei after I have shown you the overlay. Most of you, for most people, it's still complicated. It's very complicated. The problem has been considered humanely impossible, but the models managed to do it. And just give me a sec. So what I am particularly proud and happy about is that the model that has been served now to clients across the globe has been developed. The very first version of it has been developed in Tartu in our lab. So this model has made there was a lot of different iterations of this model. This model has changed a bit. But the initial version and the proof of concept version came from us, and now it's been actually made into a real product. It's been made into real product. It's been made into deployment section, and this is, this rare moments when research in the lab becomes somebody's reality in the workflow. There was a question right there. I can repeat the questions. There will be lots of them. You will have a lot of exercise. Don't worry about that. Yeah. No, it was not about evaluation. It was about doing this, right? So if I show you again this bright field image, for a human to detect nuclei, it was, well, something that they wouldn't do. Yeah, right. Right, so how did you do it, right? You're still human, right? After all. Yeah, I think I got the question. So the question was like this. You're saying that this was not something that humans do. That you cannot really just look at this image and say, oh, here's this one nuclei, here's another nuclei. Yeah, that is true. And so the question was, if that's true, how possibly you could do it, or anyone could do it. How did you derive the ground truth? How did you derive a signal that the models used to train on that sort of problem? The secret is here, right? So this is fluorescent image overlaid on top of the bright field image. So these are actually the same cells, but treated with this fluorescent dyes, these chemicals, right? So you sacrifice these guys for the sake of science. You add these fluorescent markers, and then you take two pictures. One picture is easy peasy lemon squeezy. You can detect cells with ease. And the other one is hard and nobody can do. But they are the same cells. So you see, you can take a hard one, you can fit it into a model, and this was actually what I was showing right here. You can fit it into your model on one side, and you would expect the easy thing coming out on the other side. So this is how we solve this problem. You have to, for this, we had to know the chemical physics of all this kind of, how do you prepare microscopy images, et cetera, et cetera, et cetera. So it was a little bit of a hack. We had to do a hack. But this was a good question. So as I said, proud very much, very proud, still proud. I'm still kind of very happy about this. Many years have passed, by the way. But we have then made a slight increase in complexity. We were like, yeah, fine, everybody can detect the nuclei, the middle part. How about detecting the entire cell? Because actually the nuclei is like the main sort of center of the cell where the DNA is kept. But actually the entire cell is much bigger. And its morphology can tell a lot as well. The nuclei may not tell the full story. The cell may tell the full story. Because these cells are very social creatures. They like to crawl on top of another. They like to reach out across the whole microscopy image slide. They like to do all sorts of stuff. So that was a one notch up in complexity. And we wanted to see whether we can do it. And I have actually, luckily, have one of the co-authors on the paper that I'm going to be talking about. So we decided to build a new model that can go from, again, bright field images, this very complicated bright field images, into a map of cells, of now the full cells. And this would be an instance map, meaning that you can actually see that this is cell number 1, this is cell number 2, cell number 3, and so on and so forth. So this is called instance segmentation. Not semantic segmentation, but instance segmentation. So for that, we had to figure out two things. One is that what kind of data set we needed for this, and the other one is what kind of model we needed for that, because model was non-suitable. The one that we have been using so far, it wasn't working. So first we started with a data set. We looked at the existing data sets. There were a couple of existing data sets, but they all had certain limitations about cell boundaries and how the cells look like. So we thought that we have to, build our own data set. Why I pointed here at Ali is that he is one of these people who actually helped to build this data set. So when I said that this was humanly impossible, I meant the entire human race besides this guy, and there is another guy. They too made it possible. They actually were sitting and drawing the contours that you're seeing on this slide. So all these contours were drawn by either him or the other person. It was super heavy work. I tried to do it once and I failed. I couldn't do it. I made one image and they didn't even include it into the final data set. They were like, this is just so bad, man. This is so bad. But basically they hand labeled many of these images by hand. So you can actually see these protrusions, how cells communicate with other cells. Like this. Basically this is Ali's hand. Yeah, the two bright field planes. There is another image that we have been using, fluorescence. There is a whole complicated workflow, how they have been annotating it. It took them hours and hours and hours and hours. They created this data set. And now we are actually very proud that we could publish this data set. So this data set is public. Anybody can use it for their research. We are very happy about that. And later we started looking into a model. So our previous models have been all CNNs. This means convolutional, based on convolutions. But the recent advances in transformer models, so you all know about LLMs because there are so many talks about LLMs, but actually LLMs are based on the transformer idea of the queries, idea of the key values in queries. And it turns out that there is a way to make it work also in imaging world. So we have actually made an attempt, and a successful one, to implement the transformer inside our new architecture for image segmentation. So here we have three components, main components: the pixel decoder block, the transformer decoder block, and the mask head. So this transformer decoder block is the main component that I was talking about. So this values, keys, and queries that is so integral to transformers and LLMs as one of the applications. It's an idea that the query can be an object, can be associated with an object. Now in our case, so in language, it can be about words and sentences, but in images, this is about objects, visual objects. In our case, this is actually about those cells. So every query becomes associated with a certain cell, and it kind of refines itself across the architecture until it becomes very clear and kind of represents the actual boundaries of the cell throughout the model. We have run lots of comparisons. We have looked through all these data sets which I have shown to you in the previously LiveCell, AVCon, ISBI. We have compared our model to state-of-the-art segmentation models, and we have seen that it has performed best across all of these different challenges. We have also, of course, looked into performance of all these models on our own data set that we have collected, and obviously I wouldn't be showing you this slide if it wasn't working great. It is working great. Yeah, these are the examples of how different models predicted the same cells. So you can see ours is the last one before the GT. GT stands for the ground truth, so this is how it should be, how it should look like. And you can see the approximate scores, so you can see that this value has to be as high as possible for the model to do a better job. This has resulted in a paper in CVPR workshop. It's been a nice paper. There's a QR code for the GitHub. You can scan it and you can give your star on it. The students, including Ali, who is sitting right here, will be extremely happy and grateful if you do it like this. I can see some people actually doing it. Thank you very much. This was a special request from the main author, who is actually a Ukrainian student who has worked with me for all these years of this Russian invasion of Ukraine. He was supposed to come in 2022 in February, but couldn't come because of some bureaucracy, legal issues. So we had to postpone his arrival, and this arrival so far never came because, you know, the war started. Coolio. No, I'm giving last seconds of the, yeah, a question here.
No, the QR code does not work.
Oh my God. Gosh, you can just Google the AI unit and it will show you the right GitHub. I maybe have created QR code and I haven't really saved it in the system. Nah, stupid. Sorry for that. Yes. Oh, okay. So here, this part. What I also wanted to tell you is about how sometimes, and this is more general idea that I wanted to bring up, is about using data that is normally ignored, that is normally kind of it's considered to be useful to certain very narrow applications, but it's not usually put into learning. So here I'm talking about parameters of the imaging experiment. So this is something that often comes with the images. It comes with images such as now things like microscope type, the cell line, the magnification factor. There are many things that are called meta-information that come together with the experiment, but very rarely used inside the experiment. People usually discard this information. And it's like this in many fields. It's not just the imaging field. It's across the board. Many times we only think about this main thing that we have data about, but not this additional factors that come in together with the data. So what we tried to do here was to actually incorporate this meta information into a learning, into our model. And it turns out to be very useful. So we used a mechanism called squeeze and excitation mechanism. It's basically, I'm not going to be going into all this technical detail, how exactly the squeeze and excitation work, but in essence, it's a way to ingest this metadata into a learning process, into basically models training, to support model training with this additional information. And what we have seen is that it's not only improves training, but also it can systematically alter the behavior of the model. And I'll show you now how exactly. So if you look at the top row, right? The left image is of the bright field image. And you can see with the dashed line, these are the outlines of the nuclei that you can see on the bright field image. Now, in the middle, you can see model's prediction when it has no access to meta information whatsoever. Blue and red, this means different types of mistakes. I think red was the false positive prediction and blue was the false negative prediction. So it has to predict it there, but it didn't. Now, the right image shows the model that has access to the meta information. And you can see that it actually worked slightly better. It has slightly improved performance when it has access to this meta information. But what is maybe even more interesting is the bottom line. The bottom line shows how models' behavior changes with different meta information being added. For example, the leftmost image is the ground truth. This is the huge kidney cancer. You can see huge kidney cancer in white. Now, the predictions come after this image. So the second image from the left, green contour, is what happens when you tell the model that the tumor is large. It's the meta information that comes from outside where it says, large tumor, expect large tumor. This is the prediction you get. Then the orange one is when you say it's a middle-sized tumor. And the red one, or the pink one, sorry, here, in this case it's pink one, it's when you say this is a small tumor. So you can see how models' behavior has been altered based on the meta information that has been provided, supplemented with the actual image. We have seen very similar things to style transfer as well. You could actually say now it's been drawn by Van Gogh, and the image would change accordingly. So there were a lot of interesting things happening, and this paper has actually been published. Yeah, so now small change of kind of direction. We go into from the detecting cells. I wanted to show you a little bit about how do we know which life cycle, which stage in life they are in, because that can change the treatment, that can change the way how we analyze the cells. So we need to understand in which time of the life they are in, because cells undergo a cyclic life cycle. There is a life cycle of the cells. They divide and then they divide again and so on and so forth. So we wanted to build a pipeline which looks at the bright field image and can predict the life cycle stage of the cell. There is a very interesting paper that does that from the fluorescent images, but we wanted to do it with AI, but from bright field images. With this fluorescent images, we were able to use that paper to extract the life cycle stages of the cell by actually looking at the values of the pixels inside those cells. So we were able to say, if those pixels are that bright, this cell must be undergoing the division cycle or phase. Or maybe this, if the pixels are that dim, then they are not dividing, they're in this kind of intermediate stage. Then we built a bunch of workflows. We built a bunch of pipelines. For example, we have tested a workflow that extracts certain features from those cells. So we looked at every cell at a time. Then we extracted tons of different values about those cells. For example, how large they are, how small they are, what's their circumference, and so on and so forth. Then we would put it into classical machine learning algorithms such as XGBoost or this sort of random forest algorithm, and we try to predict a cycle, the phase of the cell. Then with every other new pipeline, we try to take parts of this previous pipeline away and automate it fully. For example, with the next pipeline, we decided not to extract features, but let the AI model to figure out the features itself. So we fed it into a classification model that has predicted the phase of the cell. Now with the last one, we have decided to try to give it the entire image as an input and basically ask for the cell phases on this image, instead of actually extracting every cell ourselves. The results were we've done it, of course, for the bright field images. The results were that this patch classification has worked the best. So when you extract the patch, when you extract the part where you can see the cell and you put it through the classification model, it has worked the best. But we also have seen that this sort of like the full image prediction of the cell phases has been very promising, and we are still kind of working on that technology. Yes. I started to think that I'm actually going a little bit out of time, and I wanted to still to cover some organ-based research. With cytopathic effect, cytopathic effect is this sort of like visual features that start to show up when a virus infects the cells. So you can see it on the slide here, the small harbors, the small hubs. These are hubs of infected cells. They start to cluster, they start to get together, they start to become very dense. So these things are called cytopathic viral effects. This is when the viruses enter cells, they take over, and then they start to clump together, they form this sort of hubs. So our task was to figure out whether there is a cytopathic effect among the cells or no. We have had this sort of like large data with different amounts of virus present on the wells, and we were trying to build images, basically an AI model that can differentiate between images that have a lot of this effect, a lot of virus, just a medium hard, so there were some virus but not a lot of virus, and then those that had no virus. This is important for viral research, as we have come through the COVID pandemic, this sort of detecting viruses, being able to figure out whether cells are infected by a virus has been very critical research. That feeds into that sort of research, being able to figure out whether virus is in the cells. So we have built a bunch of pipelines, including classical machine learning, convolutional networks, transformers, trying to predict whether there is a CPE or no CPE, so cytopathic effect or no cytopathic effect, with various degrees of success, with best models being the CNNs, because transformers, as you know, LLMs, for example, they are trained on vast amounts of data. We didn't have as much data as required to feed those transformers, so CNN models usually are a bit more moderate when it comes to data consumption, so they perform better. Then we moved on to actually a different task. We decided to localize CPE, not just to say that there is a CPE or no CPE, but also to localize them. And of course, for this, we had to build detection models, and we had to label data that you can see here. So we had to, again, go down, roll our sleeves, and actually draw the places which contain CPEs. And this has resulted in a number of different ways how we can do it. One is that we can try to understand how the classification models look at the images and figure out whether there is a CPE or no. We have also built segmentation pipelines that just try to segment the CPE. And we also built object detection pipelines that try to predict objects, find objects, meaning CPE, and also segment them in parallel. So these are different classes of approaches that we have tried. the best approach was segmentation model that has worked wonders in trying to find CPEs, especially on the so-called easy cases when there is a lot of viral effect, which is obvious. And it didn't work as well on the hard cases where the CPE was barely present, but that's because of the visual similarity with the empty wells. Right, this is how they look like. This is what the XI is the explainable AI approach looks like. The YOLA is the object detection, and the right side is the UNet. Right. Now here I should have the amazing, awesome animation that will hopefully steal your attention for a few seconds. What the hell is this? This is a novel, well, relatively novel way of modeling cells. These are called spheroids. Why this is a novel and cool way is that usually biology labs or labs in biology, they have been cultivating cells on a flat surface. You might be familiar with the term Petri dish. So Petri dish is a flat kind of glass thing where you grow cells. The problem with this flat surface is that cells do not grow flat in the wild. They don't really like flat surfaces. They always grow in 3D. They somewhat grow in 3D. And spheroids is a model for growing the cells that is 3D. So you can see it has depth. You can go through it. There were many problems associated with this. However, it has enabled biologists to grow the cells in a more reasonable ways or natural ways. It has offered a bunch of challenges. For example, with this spheroid, if you look at the later slices that go a bit deeper into the spheroid, you will start seeing the degradation of the light. You will start seeing that the light somewhat scatters and becomes blurry. You can't see really that well cells on the dark side of the moon, right? So this is what happened. And in order to conquer this problem, we set out to de-blur the dark side of the moon. So the question was, can we trace the light coming from below the spheroid all the way across the spheroid so we can actually see the cells on the other side? So that was the task. We have, and actually this is again, I'm saying we have, but rather I would say that this is general research trend, is that many people have used models called GANs, generative adversarial networks. They're pretty old by now, but they're still quite useful. So this one is particularly called vox-to-vox. Why is it called vox-to-vox? It's because it takes voxels, these are the pixels in 3D, and turns this into another type of voxels. For example, you can take a brain scan and turn it into a map where you can find lesions in the brain. This will be, again, the same voxels, but they will be black and white, with white voxels representing the lesion location, while original scan contain brain scanner. So this one is called vox-to-vox. It feeds on bright field images, for example, or blurred images, and it tries to give you back the de-blurred or crispy images, or fluorescent images. So here are the predictions of this model. Every time I show this, I run into a trouble, every time. And the trouble is all the same. But I kind of remember my answer to that trouble, but the trouble persists, is that I don't remember which one is generated and which one is real. I kind of, with time, after giving many of these talks and running into the same trouble, I started to think that the middle one is, wait, give me a sec. Yeah, the middle one is the real images, and the right one is the generated ones. That has taken me a bit of time. And I will not tell you exact clues how I know that, but you have to trust me. So there are different data sets. There are 3 data sets. Going from the top is the neurites. Neurites is the connected neurons in our brain circuitry. So these are the data set of the connected neurons. The middle one is the fungi data set. This is where Syngenta, somebody asked about Syngenta. This is their data set. This is their kind of baby. And the bottom one is the cell monolayer. So this is the basically cancer cell line. grown on a flat surface. We have done a lot of different analysis. So yeah, just to maybe reiterate again, how cool is that? You go from bright field images to something that reminds of fluorescent images with no chemicals, no money extra besides your electricity bill, and no human interventions whatsoever. You just feed those bright field images that previously were considered to be crap into a model that gives you a beautiful fluorescent images back. This is insanely cool. And that's why this whole thing is very cool, is that you can actually get something out of almost nothing. And we have been looking, we weren't sure that we are not hallucinating some of these things. So we have been studying these predicted cells under a microscope. We were really looking into miniature details, like you can see here, just checking that indeed the details are there and they're real. So on the left side, you see a real fluorescent signal that has really been done. And on the right side, you can see models output from a completely different modality that has never seen fluorescence. It only seen bright field stuff. And you can see how remarkably similar the reality versus what has been generated, how do they look like. These are all 3 data sets, and they all look pretty similar. We have also done some quantitative analysis, so we have actually correlated pixel intensities from one to another. Yes, there is a question, right? So the question is that it seems that the GAN is looking for pixel differences because it doesn't have any other data. Do I understand it correctly that there is no fluorescent data on the bright field? Yes. There is no fluorescent data is an input to a GAN, only the bright field data, yeah. Okay, well, there is one but here. The thing is, GAN has, in a way, seen, but in a way that the fluorescent data has been used as a guidance for whether it's real or not. So there is one part of the GAN, which is called discriminator, that had to distinguish between generated images of this fluorescence versus reality. So this is where GAN has touched fluorescence. But when you're doing inference, when you're running the model, when you're generating images, this is where no fluorescence touches the model. You don't need, you don't actually need pairwise, yeah. There are GANs that do not require exact pairs. This one actually requires. This walks-to-walks architecture does. But there are architectures in which you don't really even need the paired versions. Okay, good. So quantitative analysis, we have actually looked into correlating pixels from reality versus generation. And we have seen there is a very good correlation between real pixels, real fluorescent pixels, and what we are generating as part of the GAN. As I said, we have been looking into spheroids. So again, deblurring the spheroids using the same GAN architecture. And we have seen that our generated spheroids, maybe you can look at the bottom row here. So at the left, you can see the input into GAN. This is how the input looked like. And you can see it's like darker, right? And these panels, they show you the depth, they show you the cut of the spheroid, either this way or that way. So it's sagittal or coronal for those people who know what these words mean. These are different cuts from different projections. Now, what you see from those cuts on the left is that these parts are dark. This part is dark, right? And in the middle, what you see in the middle here is deblurring done by a classical algorithm that has been developed separately from us. And what you're seeing in the red square here is missing cells. The reason why you're seeing missing cells is that this classical algorithm uses certain masking. It masks cells. And that's why there are gaps that should not be there, actually, because if you look at this side, if you look very carefully and very long time, you will see that actually cells are there on the background. And model does not have this bias. It just looks at what it looks, and it tries to generate a better version of it. And you can also see that these parts which were dark are now much clearer, much brighter. So this has worked remarkably in this spheroids cases. Also, these are the tumor cases. So basically, there are filled spheroids and there are non-filled spheroids. So this is a bit harsher problem when it is filled. We have seen that there are some improvements in that as well. I will actually go to tissue right away. Because I have spent a lot of time talking about cells and I have worn you out. So now it's time to shift gears a little bit. So with histopathology, with biopsy, with pathology slides, one of the biggest bottlenecks of the entire research in this area is that it's very, very hard to get ground truth. It's very hard to get people to actually manually label those images. Sitting and drawing these boxes around cells on images of thousands of pixels across is enormous work. So what they usually do when you give them image and you say, guys, pretty please, can you nicely annotate this? Very, very nicely. What they give you is this, with gray stuff being left out. When you tell them, but are these cells? They say, yeah, these are cells. But why didn't you annotate them? Well, because there's just so many of them. How are you supposed to annotate everything, right? So what we wanted was this, right? Because what happens if you train a model on the images that are in the middle? They start getting confused. They're like, what the hell? You're telling me that this blob here is the cell, and that exact the same blob, but a little bit like few pixels away, is not a cell. This is just crazy. Models go bananas when they look at that sort of stuff. So we have been thinking, and there's a lot of research done on top of on this topic. It's called sparse annotation topic. It's very vibrant field because basically in medicine and biology it's very common. It's a significant problem. We have been thinking about what are the ways how we can still use that input and get decent results back. And that's been, it wasn't easy at all. So what we have come to realize is that the way how we normally train models is we punish them for doing wrong stuff. And in that sort of case, wrong stuff is predicting cells where there are none, right? This is called false positive. This is a very bad thing. You shouldn't do it if you are a model, right? We don't appreciate that at all. We are being very rude with them when they do that sort of stuff. But not in that circumstance. In this circumstance, when the model is very confident that there is a cell, but there is no ground truth label to confirm that, we have decided to be very lenient and say, you have done well. I think it's just, it's not your problem, it's the data's problem. So what we have designed is something called generalized background recalibration loss. This is a learning strategy that says, hey, sup, if you're very confident about false positive prediction, it's not your fault. Don't push yourself, don't blame yourself. It's actually nice and fine. And actually, it turned out to work just nicely. So when you build the model this way, and you use this recalibration loss to penalize only inconfident false positives, but not to penalize confident false positives, you get better results. So for example, here, if you look at these results, the ground truth, so when you look at the ground truth, right, this is the first column. This was the time we were like, guys, but we are now very serious, like trying to annotate every bloody single cell. Thank you. And they did their best, right? The middle column is what you get with the baseline model that does not have the generalized recalibration loss. So the one that has been punished every time it would predict a false positive. And then no matter how confident or inconfident it was. And then on the right, you can see the model that used this sort of like, let's say, a bit more humane approach. And you can see that this model not only predicts a lot more cells, but also it predicts cells that have been missed in the ground truth. Because we were allowed it to be more, explorative and to be more forgiving to itself. So that has also been published. You can check it up. And finally, organ level research, so going to better medicine. So as I said, we are building a co-pilot, right? So we are building a radiology co-pilot, so something that can help radiologists, like a safety net, second pair of eyes, whatever you call it. So something that can look at the scan, find the pathologies, report it to radiologists, get the feedback, correct it. We not only find stuff, but we also measure findings, because that's what radiologists do when they detect something. They have to understand how large this something is. And also we are generating automatic reports, meaning that instead of them writing by hand or typing whatever things come to their minds, we're actually generating a formalized, standardized reporting document that will be read by humans across the globe, and everybody will understand more or less the same thing. While currently it's done that every radiologist is an artist and they write whatever they like, which, needless to say, sometimes is very confusing, even to radiologists themselves. So as I said, we have moved on since kidney. Kidney was our very first certified model. We are now closing in on the lung model that can find nodules in lungs. We are also working towards pancreas and liver, but there are other models in the pipeline about lymph nodes and bones, because these are, and stomach as well, because these are relatively frequent places where either cancer appears or where it metastasizes. So it can be either way or the other. It's either a source or a target of the metastasis. Very quickly about the kidney model. So the left image is an input to a kidney model. The right image is an output where it says that this is a kidney. This is a benign cyst. So this is something that shall not metastasize, or likely not metastasize, and does not harbor any dangerous potential to kill a patient. And the left thing, or the red thing, is the potential metastasis, or let's say malignant tumor that can metastasize. So once it finds it, it breaks this down. It measures those lesions, because they're all lesions, and it presents it in the report. I have actually designed and carried out a clinical study. So we have actually had a clinical study in our Tartu University Hospital where we have basically recruited six radiologists from the local hospital. They have been reporting those kidney studies with AI and without AI, basically by themselves and with the help of the model. We have been looking at three things primarily. First of all, what is the time that it takes for them to detect, measure, and report those lesions? Secondly, what is their precision of doing that? Do they miss things or do they not miss things when they use AI? And finally, do they agree with each other? Do they agree more when they are not using AI or the other way around? I don't have a slide with the actual results present, but basically I can tell you that, first of all, we have seen a reasonably significant, well, actually it was significant, was reasonably like big improvement in terms of time. It was 33% on average, it was faster using AI versus not using AI. But unfortunately, I don't have this slide, but basically on the slide I had a figure where you could see how during the course of the study, some of the radiologists were coming from taking a lot more time when using AI, comparing to themselves not using AI, to being much faster with AI. So at the first, when we started, everybody was very, very conservative. They have been double checking, triple checking, quarter checking everything. They were like, I know this thing is gonna trick me, and it has to be some sort of problem. So they were going around and they were spending like a lot more time when they were using AI versus when they were not using AI. While later, when they basically saw that the thing was doing the right thing, they started to just, click and done, click and done. There are certain discussions around how good or bad this is, but at the end of the day, it has plateaued around 33% on average improvement, and maximum was almost half the time that was saved by the model, comparing to usual workflows. Give me a sec, I'll just finish my thought and I'll get back there. Precision-wise, they were detecting correctly, but insignificantly. So statistics, when you do statistics, you either have a significant improvement or your improvement is merely a note. So this one was just merely a note that their sensitivity with AI was slightly better comparing to no AI. So they were actually able to find more lesions when they were using AI than they were not using AI. Similar, on a note, their specificity dropped a little bit as well, meaning that they were overcalling cancer comparing to when they were not using AI, meaning that they were likely to give false diagnosis. But again, the difference between was less than 1%. So it was very small. Question from the audience. Just tell me, I'll...
In this compiled system, does the system give only the classifications, but or also the probabilities of the classification?
Yeah, right. So the question is, when the model is this, for example, kidney model, when it detects the cancer, does it provide also the likelihood of that being the cancer? So currently the answer is no, it does not. It's unfortunate, but it's a segmentation model. So it hasn't yet been adopted to give you probability scores. We are aware of that sort of problem, which I would say problem, and we would love to include that as well. So we are kind of working on adding that feature. So currently we don't have probabilities. So the study is done, and it is about to be published in hopefully soon. And I will be very, very happy. We have submitted it to Nature Communication Medicine, and it seems that it's been rather favorable, and hopefully it will be published rather soon. Okay, one other thing which I didn't say is that when people have scan, and this scan shows signs of cancer, so lesion that has been identified. For example, here there is in the kidney there is a lesion here. It's a malignant one, so it can metastasize. So what usually happens is that the same patient is invited several times into a hospital to get the follow-up scan. Why do we need follow-up scans? Because we need to understand the dynamics of the disease. So essentially, we need to understand whether the cancer has increased in size, decreased in size, metastasized, not metastasized, etc., etc. So this is usually a very manual process that is done in radiology. They had to pull this previous scan from the archive. They have to find the same lesion again. They have to measure it again, and then they have to write a report again. So all of that sounds like, well, we can help here. So what we do is we use our model on both of these scans to detect kidney cancer again. We then do something called registration of the CT scan. So we align CT scans one on top of another, matching the kind of anatomy of a person to understand which part is where. And that enables us to compare lesions and to build the statistics around it and understand what has happened to a disease over time. And that is a very important factor in the treatment plans, so how the treatment is usually made. So they need to see whether the dynamics is going down or is it going up. That will determine a lot how the treatment plans are done. So we have actually done that, this part about registration. So aligning CT scans has been a complicated issue, complicated problem. We have done it together with, or under funding of AIRE. It's AI and Robotics Estonia Foundation. They have funded this research of detecting, of generating this sort of like deformation matrix. As you can see, this AI model that looks at both scans, and it gives you back a map of where voxels from one scan went in another scan. Because you can imagine that people in 6 months, they put up weight, they lose weight, they move, they do whatever stuff in order not to be perfect static cells 6 months before. So there has to be some work done in order to match those scans. Yeah, right. So this is what deformation matrix stands for. Yeah, this is how, by the way, this is how it looks like in lungs. So if you would do that sort of stuff in lungs, this small arrows, these are actually arrows that this visual of lungs made of. The brighter color represents bigger shifts, bigger movements. It means that something has changed in that area, comparing to darker places. Darker places means that this part has remained largely static over time. And the brighter part is when the lesion either has grown or something, or has been removed or whatnot. So this is how the deformation matrix in action looks like. So I'm finalizing my talk now with 2 last research projects. And these ones concern more of a future, more of a long-term research, more of a long-term development. And they are kind of both surrounding the same issue. And the issue is very simple. The issue is that there is very little annotated data in biomedical field, especially in the CT scan world. There are CT scans are 3D volumes. Usually it's 512 by 512 by 512. So you can imagine the number of voxels in that space. It's 512 times 512 times 512. And that volume needs to have a very rich labels. It has to have very rich information to learn from. And this rich information to learn from usually comes from humans. And the only humans capable of doing it in a reliable way are called radiologists. And they're bloody expensive. Their time is very expensive, but there are very few of them. And moreover, they're very busy actually caring for people who actually require help. There are more and more oncology cases because the population of Europe is getting old, and this old population gets more and more cancer. And also, you know what? It turns out the radiologists themselves are humans who also get cancer and get old. And they can no longer care back for the patients who need their care. So it creates a very much, a bit kind of maybe sad downward spiral when there are more patients with less people who can serve those patients. And now you probably start getting the idea why better medicine in the first place is that there has to be a way out of this downward spiral. By the way, if you have any ideas for the second higher degree, maybe consider medicine, because they do need more people in there. But why did I start that? Getting their attention away from their patients is, let's say, ethically already a little bit gray area, right? But also it's very expensive and it takes a lot of time. So getting data to train our AI models is very complicated, very hard. So instead, we have been thinking of an alternative. And we have been thinking of an alternative in a way that, what if we use an existing data? So what if we pay attention to things that are already there? For example, in Estonia, there is this is very nice place, not only because of Tartu University, which is a great place to study and work, but also because it's a centralized clinical system, meaning that all hospitals are linked together. Maybe you might have heard this from other sources, but for us, it means that there is actually a centralized place where all imaging that has ever been done in the country since 2009 is kept. It's like piles and piles of data, it's petabytes of data, with also radiology reports existing that can be paired with those images. So what does this give us is this sort of like indirect signal that we can use to train our models. And I can show you one example that we have published recently, and it's also, again, a vibrant area of research. It's called weakly supervised segmentation. Is when you don't tell the model specifically, this is where the cancer is. You're just saying, cancer is somewhere there. We know because the radiologist who reviewed this scan said so many years ago. It's now in archive. So what you're looking at right now is something called counterfactual in painting, or COIN as we called it. Basically, this is a system of GAN, generative adversarial model, that has two main parts. The one part is basically a detective. This is this part. This is the detective, that it looks at the scan and it tries to understand when the cancer is on the scan or it's not on the scan. It's that simple, just looking at the scan and trying to understand whether there is a tumor or not. And then there is another part, which is this part. I call it trickster. This part, the only life goal, purpose of this model is to trick this guy. What it tries to do is it tries to generate a CT scan such that the detective would be wrong. And the only way how it can do it is by generating image without the kidney cancer in it. So it's a constant struggle against the two. One is trying to get the right answer, and the other one is trying to generate a picture that would trick the first one. And this is how you can see, this is how this hole appears there. So these are actual results of the study. You can see the left is what the original image looking like. The contour is in red. This is actually a kidney tumor. And the middle is the counterfactual image. This one is the generated image by the trickster who tried to trick the detector. And the right image is the difference map. So you can now see how you can use just a high-level image label to understand where exactly the tumor is on the picture. I'm seeing that Jaak already took the microphone, so I better wrap up. This actually is a wrap up. The last model that I wanted to show you is the latent diffusion model. It's a model that can generate these images. So another idea that we were having is that what if we generate our own training data? Why not, right? And here you can see a couple of examples how this latent diffusion model works. Is the left, the original image, with an overlay of this white stuff. This white stuff is a place where I want the tumor to be generated. I'm saying that put the tumor there. The middle is how the model's generation look like. This is how it attempts to impute the tumor into a patient. By the way, this is like from the title sounds very horrible. You put the tumor into a patient, and usually you try to get it out. But the right column is actually the real patient scan, so it has no tumor whatsoever. Everybody's fine. So we were generating this data. It's also been supported by one of the European grants, which is now finalizing. We have been looking and building this sort of latent diffusion models. And yeah, that's basically it. We have gone through the cellular level, tissue level, and organ level research that we do, but also other people do in biomedical field. And this is all thanks to these great people. Thank you very much.